Good morning, students. Welcome to the anatomy department. And my lecture will be the first in the semester for you from our department, and this is a histology lecture. You know that our department, the anatomy department, teaches many different subjects. With a common name, we call them the anatomy. So, as you see, the histology and the embryology is also a part of the big anatomy. My name is Judit Horváth, and we will meet yet later in the semester with the embryology lectures. On this picture, you see a painting. This painting you may see also on the university between the first and the second floor. It has tiny little lights also in it. It's a beautiful, colorful painting. But this is actually the replication of a histological preparation with the so-called rainbow technique. And these are neurons of the hippocampus, a part of the brain. I hope as you pass by, you will examine these also in the original size. And another artistic product in the university is this mosaic. Please pass by as you study anatomy and histology and try to discover that what you see on these figures here, a lot of anatomical structures and also many histological structures. I hope that you will recognize more and more as you continue your studies. If you have problems with the group assignment, please contact Balázs Gassner. Here you have his email address. He will help you to find a group, or he will allow you to join a group. I would advise you to join the same group. If you do both anatomy one and histology one, then join the same group because it has advantages. If you have the common teacher many times or the common people in the group, you may help better each other. Also, please note that you must register for anatomy 1 and histology 1, histology and embryology 1 parallelly. If you don't register for both of the subjects, then you cannot do just one of them, except if you did one of the subjects, you passed one of the subjects already earlier. Please control always in the Neptune what you registered for and whether your registration was valid or not, because we see that sometimes problems happen with this. You think that you did something, but you forgot to click the last button, and then your registration might be not valid. As I told you, the big anatomy or anatomy department teaches anatomy, histology, embryology, and neuroanatomy. Every one of us will teach all of these subjects because these belong together. And also in the exams, it might be relevant to ask an anatomical question in the histology exam or the opposite way around. So please study this so that in both cases, in both subjects, you know the relevant topics, and you can apply your knowledge also in another field. The general medicine students, they study histology and embryology with these codes. The dentistry students will study in this semester only histology. These are the codes for it. The course director for these subjects is Andrea Lubitsch. The embryology part of the material for the dentistry students, it's offered in an elective course. This is that elective course. For that, I'm the course director. Please think about it, whether in your future you will need this or not. If you do parallelly in this semester with your studies the embryology one, then if you want to be a craniofacial surgeon later, or you just want to turn to general medicine studies, then you can get credit for ZT1 and EM1 as SF1, because these two subjects, these cover this general medicine student subject. So, the parts of the big subject of this histology, embryology, and one is obviously the histology and the embryology. Both come from Greek words, and in histology we study about tissues. Tissues consist of cells and intercellular substance, which are specialized for a specific function. The embryology will start from the 6th week. This is wrong. It will start on the 6th week, and it's the science of the unborn baby. And in this semester we will follow the development from the fertilization, through the morula, blastocyst, and how it will turn into a fetus, the external features. the detailed development of the internal organs that will follow in the next semester. Traditionally, embryology is taught together with the histology, although specifically in the second semester of your anatomy studies, that will be in the second year already, with the inner organ anatomy, it has more relevance to the macroscopy. But that's a tradition that we teach together, but optimally you study these subjects parallelly, so it will help you. The embryology will help you in your anatomy studies, and your anatomy studies will help you to understand the embryology. We have a website. This is the address of our website, which has a user ID and a password. If you are not using this website in the intranet within the university buildings, you must type in the user ID and the password. Here you see the series of lectures what we will cover in this semester. The histo lectures will be the same for the histology and embryology students and for the dentistry students. But as I told you, from the 6th week on, every first lecture in the week, that will be an embryo lecture. If a dentistry student registers for the embryology, then we'll have in the series of these lectures, you just simply listen also to these lectures. At the end, there will be a seminar, and you may pass the embryology exam. Now, this is a diapositive what I inserted just in the last days, because the information what I have to give you in this semester is very different from the previous semester. In the previous semester, when we started, in the previous year, I mean, in the former spring semester, in the previous year, we didn't know yet that we will have this COVID situation. So we started normally in a lecture room with a lecture room lecture. But now we know that we must start with the online classes. If the epidemic situation allows, we will turn to contact classes in the second half of the semester. Unfortunately, nobody knows yet this for sure. These topics will consist of lectures and out of practices. The lectures will be either real-time lectures on Teams, or they will be pre-recorded. But in both cases, these recorded lectures will be later available on our YouTube channel. The practices will be, again, either on Teams or in the Zoom. Your group teacher will contact you and will explain you how he would like or she would like to give you the classes. You have to attend, actually you have to attend in real time these classes. Your attendance will be recorded, the absences also, and there is a limit for the absences. For that you will find also information on our website, and if you exceed this limit, your semester will not be signed and you may not sit for the exam. For your studies in this semester, you need a computer or a laptop with camera and microphone. We don't suggest you to follow these classes on a telephone, mobile phone, or on an iPad or a notebook, because it's much smaller and it doesn't give you enough details. All important information will be posted on the Institute's website, and parallelly many times you also receive an email through the course mail, through Neptune or through the Teams. So please do not neglect our letters that we write you, because we try to inform you really in advance about everything, and we would like to help you the most to complete your studies in this semester. Now about our website. So I showed you the link before, and if you click on that link, then you get to this page first where you can choose the language. Probably you will choose English language. Then you go to, with that you get to the next page where you have a general description, and you may click on this information. Here to information. If you go to this information, then this page will appear where there are descriptions of the subjects for the medical students. Now for us it will be the Histology 1, SF1 relevant. There you have a general information about the requirements. You have the list of the lectures and the list of the practices. There is a separate part where we inform the dentistry students, though the histo lectures, as I told you, will be the same for the histo and for the gen med students. Also on this page, you will find the most actual informations, newest informations. So please go to this page, try to examine it, and before you ask any questions for the course director or from anybody in the department, please try it. You might have the information on our website. Okay. Please also press frequently the F5 button to refresh the page. So if you go to this page back and you don't click on information, but you see what is here further down, there are several teaching materials here, for example, also for histology. The histology teaching material follows the weekly practices, and it's a practical help. I would like to emphasize that this is not a textbook. This is a practical help, which helps you with that what we require you to show in the actual slides. You have also small icons here. If you click on these icons, then the histological picture will be larger. These are static pictures, but they show you very typical parts of the slide where you can see the most important elements. Also, we have another technique, what we will use in the labs. These are the zoomed, the scanned and zoomable slides. With that, you will learn histology in this semester. Unfortunately, we cannot use now the real slides with the real microscopes. But you will have all the tissues in the labs about how to use that and what is that address that you will get the information from your lab teachers. Again, please press frequently F5. And we also have our YouTube channel. On the YouTube channel, we have in 3 languages a lot of lectures, a lot of teaching material, a lot of demonstrations. Among others, we have also histo demonstrations. It's not complete yet, not yet for the entire material, but we work on it and we will give you more and more on this page, so it's worth to examine it, that what is new on the page regarding the subjects that you study in the spring semester. So if we would be in the real lecture room, I would ask you the question, who would like to have a peaceful summer vacation? And most of you would like to have a peaceful summer vacation. And then my next question used to be that when does the exam period start? And when I ask these questions, then many of the students start to examine a calendar that when are the 14 weeks over and when the exam period starts. But that's not the true answer. The true answer is that the exam period starts on the first week in February when we start with the lectures and the labs. If you follow the material and if you decide that you want to pass this exam, then definitely you will be able to pass your exam, your histology exam, in May or in June. With the online teaching, we see a kind of like a polarization with the students. Those who take it seriously, those are really good because they can follow the material, they study at home, they come prepared to the online classes, and they ask clever questions, and they are updated with their knowledge. But it has also a disadvantage that we do not see who is sleeping on the other side of our computer screen, of our connection. So there is a chance to make it lazy and not to follow the material. And if you once lose the line, then it's very difficult to catch up with the material. So please consider what is your aim. You just want to have a good time in page and sleep a lot and party a lot, or you want to have a successful exam at the end of the semester. If you choose the second, then you have to start to work immediately on the first week, and then you will succeed. We wish that you would succeed. So what are the materials that you have to use for your studies? You need a histology book. This is, nowadays, probably this is the best histo book, this Pavlina book. We need an embryo book. There are several embryo books, and all of them are good, but the Langmann's book is the one that is printed in 3 languages, and that's the background of our lecture material. So we advise you to use the Langmann, because based on that book, we will give you the lectures. And also, please, buy this histology workbook in the basement bookstore of the university. it's cheaper than if you just buy a copybook. And in this histology workbook, you have a page for every slide what we study, and very simple questions what you must answer. You must answer them on your own. Of course, if you have problems or you have some doubts that you didn't know the right answer, then ask your teacher, your group teacher. And there are also lots of empty spaces because we would like you to draw the slide, what you actually examine. So please have some colored pencils ready, examine the slide. It's not that you have to copy a particular part of the slide what you see on your screen when you zoom in. It's that you have to draw your impression that what is the actual build-up of that tissue. And if you do this work in your brain and you are able to draw, then that will stabilize your knowledge and it will help you to pass the exam. Besides these books, you may use also our internet material. As I told you, we have it on our website and we have it also on the YouTube channel. Plus, if you want to look after something, you may use any internet source, including Wikipedia. Usually the Wikipedia information is quite good, so it gives you help. Just please try not to get lost in the many pieces of information. So find the guideline, and the guideline should be the lectures, the labs, and the textbook. This should be the guideline, and everything else just helps to stabilize that knowledge what you get through these major channels. The histology workbook, as I told you, you must fill it in, and at the end of the semester we will control it. If you didn't fill it in and you didn't draw the drawings, your semester is not accepted and you may not sit for the exam, so please take it seriously. Okay, and once you return to the university to real classes, then please also have your names on your books, because many hundreds of students have similar books, textbooks. Okay, so why do you have to draw? With these drawings in the classroom, when I give the real live lecture, I used to ask that, what do you see on these drawings? And what a miracle, everybody realizes or recognizes that this is an apple and that's a pear there. But if you look at this, then here it's not really like an apple. The color is not like an apple. The apple is not angular, it's not a line, and the pear is not blue either. But you recognize it because you have an experience how these objects look like, and you conclude as a simple drawing, from a simple drawing, that this must be that object. If you think about children with the age 3 or 4, they can draw a dog, which is not a Rembrandt painting, but it's obviously a dog. So you don't have to be a Rembrandt, so nobody should tell me that I cannot draw, because you could draw as you were a child, 4 years old, you could draw a house or a dog or your family or whatever, and everybody recognizes that that's a dog and not a giraffe or an elephant. So you have to be able to draw at this level, and you have to, and if you do this brain work to conclude it to this simple to that complicated tissue to a simple drawing, then that means that you understood the structure of that tissue. Okay, a short history about histology. First of all, the microscopic, it's also called microscopic anatomy. Why? Because anatomy started as the macroscopic anatomy, things what you could see with your bare eyes. And people were curious and they wanted to see always more and more. And at one point they got to that level that they had to use a magnifying lens. And then they figured out how a microscope could be designed. The first microscope was designed, he gave the name at least, Faber. He made that microscope for Galilei. In science it happens many times that that scientific knowledge comes to a certain level, and then in several parts of the world they discover the same or similar things because the science got to that level. In the 17th century, science got to that level that in many regions of the world they discovered the microscope. So among others, this Faber, he also made one. Robert Hooke used also this microscope to describe the minute bodies, which became a scientific bestseller in the 17th century. And he gave the name cell for these small units because in botanical tissues what he examined from plants, he observed that there are small little units, and he called these units cells, like the monks had the little cells in the buildings where they lived. And ever since we use this term cell for these little units of the tissues. And then there was Malpighi. He lived also in the 17th century, and he established the histology, what we call histology today. He examined a lot of tissues, and he designed terms what we use even today, like the Malpighian corpuscle in the kidney, or the Malpighian follicle in the spleen. So ever since these terms are used, he was the one who described this for the first time. And Leeuwenhoek, who was a merchant in Delft, he also 17th, beginning of the 18th century, he was not a scientist, but he was a very curious person. And he was told to have cultured fleas in his pocket. Because at that time, for us, now it's obvious that these bugs, they lay eggs, and from the eggs you get the worm-like thing, and from that you get the bug, and so on. So these are very obvious things for us today. But at that time, it was not really known that how the flies are born. And he was the one who discovered this, and he went after these problems. Among others, he always also examined sperm cells. That was also quite easy to examine, and made nerve cross-sections. And in the nerve cross-sections, he saw these structures, what we today call nerve fibers. At that time, he didn't know yet what that was. He just saw that there is something in it. And then at the end of the 18th century and the beginning of the 19th century, as this microscopical science developed, they started to use dyes to stain the tissues. They made thin slices, and initially they examined this not only in a native preparation with the diffraction of the microscope, but then they started to use dyes. Mostly German scientists are listed here, because in the 19th century, beginning of the 20th century, the German-speaking territories were the territories where science was most developed. And these scientists, they used dyes. A few of them we use also today, like the hematoxylin or the aniline or the carmine, azocarmine. So we use them even today. A description about these stainings, you can find a chapter on our website. Please read it, because that's also part of the material. Well, as I told you, anatomy and histology, they are really close to each other. So if you take the humerus, which is closely 50 centimeters long, the head, or this distal end piece, that's about 5 centimeters in measure, and a small thing on it, like this bump on it, this might be close to 5 millimeters, and so we can go down with these macroscopic structures to about 0.5 millimeter, a small groove with 0.5 millimeter diameter that we can yet see with our bare eyes. 0.5 millimeter, that's 500 micrometer, right? That's also already a thing, what you can see also in the microscope. And with the microscope, with these light microscopes, what we typically use, and the pathologists, in many cases, they use the same microscope, so that they don't really have to go in much bigger details many times. With that, you can go down with the magnification to about 1 micrometer or 0.5 micrometer. That's 500 nanometer. And the things that are smaller than 500 nanometer, those can be examined by electron microscopes. Electron microscopic pictures, you will see them in the lectures. Those are part of the theoretic material, and you have to be able to talk about them in the exam, or to interpret a picture. Your teacher may show you an electron microscopic picture, and you have to be able to do an interpretation that what you see there. So that's, don't neglect electron microscopic information, because you must know that also. Another thing which might be a problem, that you do not know these measures and the units properly. If you have problems with these, then please revise. Please revise that what are these, because you have to be sure that how long is 10 millimeters, and what's the relationship between a micrometer and a millimeter, and between the micrometer and the meter. So you have to know these in order to get oriented in this microscopic world. On these pictures here you see, as we magnify, always bigger, the things. So this is here about 5 centimeters, 5-6 centimeters in the transverse direction. But if we would make a ground bone, then we would see these round units, which we call osteons. These osteons may be up to about 200, 500 micrometer if they are the largest. And if we go further with the magnification, then we see here these little lines. If we enlarge them, then we see what would be the shape of the osteocytes. They look like myoepithelial, so it's like a bug body here in the middle, and they have many, many little processes which connect to each other. This body, this is about 10-15 micrometers in transverse direction, and the processes may be up to 20 micrometer long. And if you go to electron microscopy, then you see the cell body of these osteocytes, which are residing in these lacunae, what you see here. It's worth to click on this link, find this link on the internet, because that guides you through these sizes from the coffee bean to the hydrogen atom, that how these are related to each other. So it gives you a good impression that you are not lost. You know, the problem is that a cell or a nucleus or a mitochondrium or a ribosome in your biology book or even in a histo book, when they are drawn, they don't show the actual differences. It's there in the text that how big they are, but as it appears for your eyes, you don't see the difference in size between a ribosome and a cell. And we see this many times, that you have problems with this, judging the sizes of these structures. You have to be able to judge this. And I give you now a series of examples for that. So if I show you a stone or a nail, and I don't give you any other information, then you have no idea how big these are. Because it may be that the stone is smaller, the nail is larger, or they are about the same size, or it's the opposite way around. So you cannot tell unless you put them next to each other. If you put them next to each other, then you know that the nail is twice as long as the stone, right? But you still do not know how big they are. They might be both big, or they might be both small. And in order to find out that how big they are, you have to compare it to something that you know how big it is. So that's my hand. This photo is about 20 years old. So that's my hand here with this nail, with this stone. And here the students usually tell me that, okay, then this piece of stone is 3 to 4 centimeters in diameter. And in the old times when I made this photo, then my next question was that which picture could have Leonardo interpreted? And then you usually say that, okay, the one with the hand. But what happened? 20 years ago, this was a cell phone. And these cell phones were about this small, about 8 centimeters long. But nowadays, you don't know that how big they are. At one point, I thought that I would change it for something else. But then I thought that, okay, this has some information, that the newest thing on this photograph, that is the most outdated. Right? So now you got back to the point of Leonardo. You can interpret this picture, but you have no idea how big this cell phone was. And if you have this information that, okay, the stone is about 3 to 4 centimeters, then from here on, it's all the same whether you show this on a big picture or a small picture. In your mind, you will have also this real size, the 3 to 4 centimeter size of this stone. Why did I tell this all? Because in everyday life, you use this knowledge. How do you use this? That if you see a picture of a little girl, this is Nono, and at this picture she was about 140 centimeters and 25 kg. And if you see this picture, regardless how big it is, in the lecture room this could be really big, it could be 3 meters. You wouldn't have thought that this girl was 3 meters tall. You wouldn't have thought either that she was 3 centimeters. You would have been able to judge that she is somewhere between 120 and 140 or 50 centimeters. Right? So you have an everyday experience, what you use in your everyday life. You must get to this point also in histology. So if you see a histological slide, you must find familiar objects. Like, for example, a lymphocyte should be a familiar object, this little blue ring here, or dot here, which is a small lymphocyte. It's about 5 to 7 micrometers in size. And we can compare to that. knowing that this is 5 to 7 micrometers, you can judge that the height of this epithelium is, let's say, 60 to 80 micrometers, right? But you you shouldn't tell that it's 2 micrometers, and you shouldn't tell that it's 500 micrometers. So you must be able to judge the relationship in size between the histological objects, and for that you need these familiar things like a lymphocyte, an erythrocyte, or a fat cell. And it's also, you see, this picture is colored, this one is black and white. Well, until about 30 years ago, the colored printing was yet quite expensive, and the technique was much more expensive than the digital technique nowadays. So most books, histo books, they had just black and white pictures, also from the H&D stained slides, and everybody could interpret them. Also for you, it wouldn't be a problem that know whether the skin of Nono was gray or kind of like skinish pink. You look at this picture and you think about the pink girl with the blonde hair, although here everything is now black and white. It should be the same story with the slide, regardless of the staining and regardless of the photo, whether it's black and white or colored, and regardless if someone has problems with recognizing the colors well, you have to be able to recognize the structures, and you have to know that with that staining what should have what color. You have to know that if it's an H&D staining, then the nuclei should be blue. So this is also partially a theoretical information. It's not just what you see with your eyes. Okay, so what yet about our slides? So the thickness of our slides is usually between 5 and 10 micrometers. Rarely, mostly in the nervous system slides, we use also thicker slides. The electron microscopic slides are much thinner, but as I told you, we show you those only on photographs. Most of our slides are dyed, but occasionally we will use native preparations, like with the pigmented epithelium, which contains melanin, or you can see the color of the melanocytes because they have the native color. Where do our tissues come from? Anatomists and histologists in our department would like to show you healthy, normal tissues. So if we show you the real tissues, the real slides, sometimes we have to look for a long time for a good specimen. Sometimes we examine 20 tissue blocks till we find one and tell that, okay, now this is good and we would like to use this in teaching. Where do they come from? They may come from cadavers. Of course, these cadavers have to be relatively fresh. Rarely they come from animals. We don't really like to use animal tissues for teaching you human histology. They are very similar, but still. So if we teach you human, we would like to use human tissues. And sometimes these human tissues, they may be obtained during an operation. So if for some reason they have to remove a part of an organ, then from the healthy edges we can get such tissues. Pathologists, they don't have a choice. They have to examine whatever they get, so they cannot tell that, okay, I don't like this cyst, I would like to examine another cyst, because they have to examine what they got from that patient. It may happen that if they look for a tissue block what they use in teaching, then they will choose from the possible variations that one which is the most beautiful, in quotation mark. Right? But they may not tell that I don't like this heart infarct, I would like to have another one, because that is already another patient. So for the diagnosis you have to use that specific tissue what you get from that specific patient. Okay, so ever since the 19th century we have distinguished these 4 basic tissue types: epithelial tissue, connective and supportive tissue, muscle tissue, and nervous tissue. And please learn them in this sequence. The sequence is important because they follow each other always in this sequence. So on the surfaces you always have epithelia. Under the epithelia you have always connective tissue, no exception. And beneath these 2 layers, in most cases you have muscle tissue. And the nerves, they may go everywhere. So they will find every little piece and they innervate the structures. In these coming pictures, I will give you a very brief introduction into histology, only the very basic characteristics of these 4 basic tissues. The details will come in the following weeks in many, many lectures. So what about epithelial tissues? We have the surface epithelia. They may have different shapes, and they may be stratified or not stratified. And we have, as another big group of epithelial tissues, these will be the glands. The glands will open always to an epithelial surface, or almost always. And the epithelial tissues have densely packed cells. They are closely attached to each other, and they are avascular. So there are no blood vessels between the epithelial cells. These are the most characteristic features. Connective and supportive tissues. They always consist of cells, fibers, and ground substance. On every page of your histo word book, you will have these questions when you study one specific connective or supportive tissue, that cells, fibers, and ground substance. And with every one, you will have to list that what are those elements. Always the cells are the most important, because without the cells, you wouldn't have the other parts. But it may be that not the cells are dominant, but the fibers or the ground substance. And whichever part is dominant, that will give the specific characteristics of that tissue and also the physical characteristics. On this picture here, now you see a loose connective tissue, very picturesquely drawn with the fibers and with the cells in between the fibers. And this is here a drawn model of a bone where you have these round units, what I already showed you previously, the osteons, right? And you would think that, okay, where are the fibers here? Where the fibers are in these lamellae, in these special lamellae, eversion lamellae. These will be collagen fibers, and they are embedded into a material which is mostly different calcium salts. So that's why that covers kind of the fibers, and you don't see the fibers, but collagen fibers are there in the bone. Now, muscle tissue. The most characteristic feature of muscle tissue that it may contract. We have smooth muscle, heart muscle, and skeletal muscle. Both the heart muscle and the skeletal muscle are striated muscles. Here we have a problem with the namings. Nobody has ever in any language found out a good name for these muscles, because mostly when we speak quickly about these muscles, then when we say striated muscle, then we think about the skeletal muscle. It's not a mistake if you think about that. Just you must know that not only the skeletal muscle is striated, but also the heart muscle is striated. And then we have the nervous tissue. Well, with the nervous tissue and the nerve cells, everybody thinks that that's something very small and delicate, and you have to be very careful not to destroy it. Well, the nerve cells, nervous tissue, first of all, consists of 2 big cell groups, the nerve cells and the glial cells. And the nerve cells, they may be small or they may be extremely large. So the biggest nerve cells, the pericardion, is maybe about 100 or 150 micrometer in diameter. That's huge. It may have long dendrites, several hundred micrometer long dendrites. And it may have an axon, which may be over 1 meter. Of course, not all nerve cells have such a long axon, but those which run to your toes, for example, these are over 1 meter, because you have the cell bodies on a much higher level. What is the function of this nervous tissue? It receives, analyzes, and reacts to the impulses what you get from your neighboring world. Sometimes the tissues, they have kind of like transitional features. If we speak about fibers, then we associate fibers with the connective tissues. That's good. That's absolutely good. But you must know that sometimes smooth muscle cells may also secrete fibers. And if you speak about contraction, then that you associate with muscle tissues, which is again correct. But sometimes the fibroblasts may have contractile elements, and then we call them the myofibroblasts. Or in the case of glands, around the secretory cells of a gland, you may have so-called myoepithelial cells. These have also actin and myosin filaments, and they may contract. Here you, for example, see a cage of these myoepithelial cells around a gland in the skin. The nervous tissues have also common features with epithelia, in that sense that both of them are polarized. Or the nerve cells and glial cells, they don't fit on each other compactly, so there are intercellular spaces, and in this intercellular space, the substance is very similar to that of the connective tissue. So these tissues are kind of separated from each other, but still are connected through these common characteristics. Each of the tissues have so-called intermediate filaments. That gives the cytoskeleton. In the case of epithelia, this is cytokeratin. Keratin is the intermediate filament. And different tissues have different intermediate filaments, like you see it listed here. These are important, especially for the pathologists, because as they show the intermediate filaments with immunocytochemical techniques, with that they can identify that what is, for example, in the case of a tumor metastasis, what was the original tissue. So this helps the diagnosis. Okay, in the following pictures, I will show you my own drawings, which summarizes the basics of histology. It might be that you don't understand everything or you cannot remember everything what I give you here, but these are basic informations, and I would like to suggest you that go through these pages several times during the semester, especially when you start and finish one big group of the basic tissues, because it might help you to understand the logic of the histology. So on the surfaces, you always have some kind of an epithelium. The shape of these epithelial cells may be squamous, cuboidal, or cylindrical, or the epithelium may be simple or stratified. What decides that what kind of epithelium do you have? It's always the function. So nature always does what is the best in that situation. So if there is diffusion needed, then you will have simple squamous cells. If the cells, they have to secrete something, then the cytoplasm will increase. Those cells which are resting, they don't act actively in that moment, they will build down the cytoplasm and the nuclei will be compacter. Right? And if it's activated and it has some function through kind of like any stimulus, then it will grow cytoplasm with cell organelles, and it will also change the structure of the chromatin in the nucleus. It will be much looser to allow the function of the nucleus. If protection is needed, then nature will use stratified epithelia. If it has to protect against evaporation, like on our surface, the surface of our skin, that will be also keratinized. So it's very, very practical as this is designed. And if you understand the logic, then you will not make mistakes with that. So please remember that no cell keeps up cytoplasm without function. Beneath the epithelium, there is always a basement membrane, right? There you see this basement membrane, which has two layers. It has a lamina basalis on the epithelial side and the fibroreticular layer. This is very important for pathologists. It's a different stage of a tumor if it breaks through the basement membrane. That's already, of course, not that good if it breaks through. So it's a very important structure, the basement membrane. It's not the only place where you have it. You will see at the end of this series of drawings that all basic tissues are separated from each other with this basement membrane. The epithelial cells are anchored to each other with junctional complexes. These may be the tight junctions, the bell desmosomes, the spot desmosomes, the gap junctions, or the hemidesmosomes. You've studied this also in biology. And please remember that whatever you studied in biology, you have to use it also in our histology material. We will not teach you one more time. We know that you studied that in biology. So we have the junctional complexes. And we may have also glands, and the glands, they always open to the surface, And the glands always develop from the surface epithelium, so I could imitate it so that you have a surface epithelium and that's pushed in like this. And then you get a gland. Around the gland, of course, the basement membrane must be continuous. Under the epithelium, as I told you, there is always connective tissue, no exception. And as I explained earlier, connective tissues consist of cells, fibers, and the ground substance. On this picture, because that was simple to start with, first I drew here the fibers. The thick pink lines resemble the collagen fibers, the wavy thin ones, those the elastic fibers, and the straight black ones imitate the reticular fibers, which is actually a type of collagen, but it's stained well with the silver nitrate. Between the cells, we have this, between the fibers, we have the cells. These cells may have two groups, fixed and mobile cells. Fixed cells are the fibrocytes and fat cells, for example. And as a fibrocyte, for example, you must not imagine it like if it was frozen. So it can stretch out a process or pull back according to the needs, but it cannot go away from that region. It can move just in the immediate vicinity. Right? The mobile cells, those are all white blood cells. Right? These white blood cells are produced in the bone marrow. They get into the bloodstream and they go to the destination where they function, to the connective tissues. So they are just transitional elements in the blood and they do their job in the connective tissues. And here you see one more cell, these black ones. These are the melanocytes. Well, melanocytes are very interesting cells, and if you look at the textbooks, they are very quiet about that to which basic tissue they belong to. I used to say that if it was me, I would connect them to the nervous tissue. Traditionally, we teach melanocytes then when we teach the connective tissue cells, but they are not really connective tissue cells. Sometimes they may be in the connective tissue, like in that eye slide, what we will show you in this semester from the choroid layer. Then there they are in a connective tissue surrounding. But in most cases, they sit between the epithelial cells, like in the stratified squamous keratinizing and non-keratinizing epithelium. These melanocytes give you the color of the skin. They produce melanosomas, they give it to the epithelial cells, and that's what you get brown when you go to the sun. The melanocytes, they develop from the neural crest. They migrate out and then they reach the periphery. If they are stuck in the connective tissue, so they don't reach the surface, if they are stuck in the connective tissue, sometimes that happens, then they have kind of like a bluish color. So that's then a spot on the skin which may have a bluish color. That's not really blue, it's just that the melanocytes are deeper and the tissues which are above it, they do this diffraction that at the end you get this bluish color. And all this is embedded into an amorphous ground substance which consists of different molecules. And here I showed also that the fat cells are very selfish cells, so they would like to separate themselves from everything else. They have also a basement membrane on the surface. That's a unique thing. Okay, so connective tissues are very important. Sometimes students, especially if you have the chance to dissect, then you have the feeling that this loose connective tissue, which is in between the main structures, that's just a stuffing. Well, it's not just a stuffing, it's very important. It's kind of like a buffer territory for the body, also with energy like the fat cells, and also with ions and water. Right? So you can, you can stand for a while without water. That depends on the water content of your connective tissues. Or if your circulation is not good, then this excess of water is deposited into the connective tissues and it appears like an edema. And not only in this loose connective tissue, but also in many other territories of the body, like in bone, cartilages, tendons, skin, you have a lot of collagen fibers. And please, skin is not only the surface epithelium, it's also the underlying dense, irregular connective tissue, right? So if I speak about skin, then I speak now about the connective tissue part of the skin. So all these parts, they contain a lot of collagen. So at the end, all of your total body protein, to about 25%, that is collagen. That's a lot. So if you think about animals, what we eat, it's the same proportion. So we cannot throw away 25% of protein of a pig or a beef or a chick or whatever. So we must use it somehow. How do we use it? That we make jelly out of it. In Hungary, in the wintertime, it's a very typical dish that we eat jelly. Some people, they like it with the skinny parts. Some people like it with meat. But this jelly-like substance around it, that's actually a frozen collagen solution. Frozen, I don't mean that it is minus degree. It will be kind of like a jelly also on 5 degrees or 8 degrees. And if you think that you don't like this dish, then maybe you eat bologna. And if you think about these cold cuts, which are in these plastic tubes, that's the same story, just in a different shape. There is some food color in it, that's why it's pink. Main part is maybe food color, some meat, and that it holds together and it has its shape. That depends on the collagen, which is added from the skinny portions of the pig. Or if you eat the Gummibärchen, you know, these little bears, or if you eat a cake with the gelée on the top, that's everything collagen. That's simply collagen. And it was also the glue for the carpenters. The term glue comes from that when bones were cooked, and from that a special glue came, which was the glue for the carpenters. It was kept warm in a pot, and it is a very good type of glue for wood. Okay, so in the deeper layers, we find muscle, which may be skeletal muscle or smooth muscle. Right? Between these two, in diameter, there is an extreme difference. Right? The skeletal muscle cells, there are 50 to 100 micrometer. The smooth muscle cell diameter, that's 3 to 8 micrometer. So it's a very big difference between these two. And on the surface of these muscle cells, we have also a basement membrane. If we talk about muscle tissue, then it's never only muscle tissue. Between the muscle cells, you always have a little connective tissue. Right? The skeletal muscle in the kitchen appears as meat. So if you buy a piece of meat, the more expensive it is, the less connective tissue you have between the muscle fibers. So those meats are most expensive, which have practically no connective tissue between the muscle fibers. You know that, you must know that from your kitchen experience. Okay, so here you have a little connective tissue, but in general, when we see a piece like this in the tissue, then we say that, okay, this is striated muscle, and we don't say that, okay, these are striated muscle fibers mixed with some connective tissue in between. The vessels will enter all these tissues, right? It comes out from the heart, it capillarizes, it supplies all the tissues, it will go near the basement membrane here, and with diffusion it will supply the epithelia. In the wall of the vessels we have also epithelial tissue, connective tissue and smooth muscle many times, but we don't say for the connective tissue that the endothelial cell of a vessel or the smooth muscle cell of a vessel belongs to the connective tissue cells. Please take care of that. We know that in the connective tissue we have blood vessels, but we don't count it with the connective tissue cells. And you also have nerve fibers. Nerve fibers may be motor fibers, somatomotor, that the multipolar nerve cell is sitting in the central nervous system, and the axon comes out. The axon has a sheath around it, and the axon plus the sheath, that makes a nerve fiber. So nerve fibers come out to the skeletal muscle. Or nerve fibers come out and they are going to a ganglion, which is a collection of nerve cells, where it will synapse with the next step multipolar nerve cells, which go then to the smooth muscle and to the glands. Right? So these are the nerve fibers, which are the motor fibers. And we have also sensory fibers, which start with a receptor. On the receptor, there is the synapse of the peripheral process of this pseudo-unipolar cell. and the central process goes into the central nervous system, and there it will be analyzed. Both of these processes are nerve fibers, that means axon plus sheath around it. So I didn't put it into the picture, but around the nerve fibers you also have a basement membrane. So if we would have a small Mickey Mouse here in the middle, then if it looks into the direction of epithelia, it will see basement membrane. If it looks at a fat cell, it will see basement membrane. If it looks at a gland, it will see basement membrane. If it finds a nerve fiber, it will see a basement membrane. And if it finds a muscle cell, it will see also a basement membrane. So on the border of all basic tissues, we always have a basement membrane. About the preparation of the tissues, these steps, you must know these steps, and about this you will hear in the labs, and you will have to know the material, what is written in this first week's material. So please read it from this chapter. Here you see some tissue blocks embedded into paraffin, and this is here a microtome with which we can make very thin slices of this tissue. These are the dishes in which we do the stainings. And what stainings we use, as I told you, occasionally we use unstained preparations where we have melanin in the cells, or iron hematoxylin. This has shades of black, so gray, black, white. And we see well the nuclei, the kinocilia, and the line of the basal bodies. Hematoxylin eosin staining, that's a very common staining. Nuclei are blue, and the cytoplasm is pink. The fibers are also pink, but stronger pink. Sometimes the shades may vary between two H&D stainings, but basically they are the same. Or we use the azon staining that comes from azocarmine and aniline blue. Here the nuclei are red. So the most important difference is that here the nuclei are blue, here they are red. Nuclei are red, cytoplasm is pinkish, and the fibers are blue. With the pus hematoxylin staining, the pus stands for periodic acid Schiff reaction. This will show the carbohydrates like the mucogen in the goblet cells, that's for example a carbohydrate, and the glycocalyx on the surface, that's also a carbohydrate. So this will appear in this purple lila or magenta lila color. The nuclei are blue because we also use hematoxylin. In this case, orsein is specific for elastic fibers. It may be combined with hematoxylin, like here, or sometimes we don't use hematoxylin, we use only the orsein. Silver nitrate, that is typical staining of the nervous tissues. It has a blackish, dark gray or brownish color. Here you see the big multipolar nerve cells, and here you see some nerve fibers running. Fats you can only stain if really staining the fat, if it's in the slide. In the normal situation, normally in the routine slides, the fat is dissolved and we get these empty rings. Right? So we know exactly that these are here fat cells, but you also have to know that fat is already not here, it's empty. It's dissolved during the procedure. You may stain fat in membrane preparations without going through the embedding story. For example, with Charlock Red, here it's actual fat within the cells, or with osmium tetroxide. You can use also frozen sections, and in this case the unsaturated bindings between the carbon atoms are oxidized with the osmium tetroxide, and you get these black rings where you have the myelin. And the myelin contains a lot of fatty molecules. This Charlock staining is based on the distribution quota of the dye between the water phase and the fat phase. Sometimes something like this you use also in the kitchen, like the paprika or curry. Right? In the fatty part of the meal, the color is stronger always. To evaluate that what you see, for that you need a theoretical background. So you always have to think about that why is that color in that particular cell. Here I showed you just an example of pituitary with immunofluorescence, with immunohistochemistry, and with an H&D staining. These are all from the same tissues. They look very different, And you have to have some theoretical knowledge in order to explain that what is what you see. And as the last picture, again a beautiful picture about a rainbow, about this rainbow picture, and here you have the short description that what is in the background. Thank you for your attention, and I wish you all the best in the semester, and I hope that you will have a successful exam in May and June. Goodbye.